Welcome everyone to today's Emerging Topics in Biomolecular NMR seminars. Today we have two fantastic speakers, but before we get started, I just want to remind you that these talks we do record at the speaker's consent, and we would like to ask you to not make your own private video recording of the lectures. After the seminars, these talks are posted on ICMRBS YouTube channel anyway. During the talks, when you have questions, please type them in the Q&A, and we'll invite you or read your questions afterwards. And after the hour, if you have additional questions, you're welcome to stay for the informal sessions. Today we have two fantastic speakers, and it's my pleasure to introduce the first speaker, Wei Qiang. Wei Qiang obtained his undergraduate degree from Tsinghua University in China, and then moved to Michigan State University and did his PhD work in David Willicke's lab. Then in 2009, he went to NIH and worked in Rob Tico's lab. And in both labs, he did some of the most significant work in each group. In Willicke's lab, he studied the HIV fusion peptide structure in cholesterol-containing fairly biological membrane mixtures, and characterized and elucidated the beta sheet structures of this fusion peptide domain. And in Rob Tico's lab, he investigated the A-beta structure. There is an Iowa mutant that he studied and discovered the anti-parallel packing, which is different from the wild type A-beta. He also examined the fibril formation kinetics and stability. Most influentially, I would say, he looked at the brain-seeded A-beta fibril structure and published very significant papers. And then since 2014, he has started his independent career at State University of New York, Binghamton, where he sort of merged his two backgrounds and has been investigating how A-beta peptide interact with lipid membrane. So they're a very interesting topic. And he has also independently been studying a low pH insertion peptide that is of interest to cancer detection. And so today he's going to tell us his A-beta work, interaction with the lipid membrane. Welcome, Wei.
Thank you, Dr. Hom. Okay, first of all, I would like to thank the organization and also the very nice introduction. My name is Wei Qiang. I'm from SUNY Binghamton in New York. And my group is interested in the biophysical and structural biology studies on the beta amyloid peptides and also other membrane-associated peptides and proteins. So today I'm going to talk about one of the main directions in my lab, which is the membrane interaction of the 40 residue beta amyloid peptides. So, okay, so this project is related to the pathological mechanisms of Alzheimer's disease. As you know, that this is the #1 severe aging problem in the United States and also around the world. And here shows the, let me turn on the laser pointer. Okay, so here shows the clinic symptoms of the Alzheimer's disease human brain. And you can see there significant shrinking of the brain volume and the time-dependent spreading of the amyloid plaques, which could be stained by multiple types of dyes. And this is the brain tissue stained by Congo red. You see those red, the amyloid plaques in red color. And if you look into those amyloid plaques, you see those filament-like aggregates, and those are known as fibrils. The major component of the fibrils are 40 or 42 residue beta amyloid peptides. And there are numerous structure studies on the molecular structure of the A-beta fibrils, and they share the common structure feature called cross-beta, which means that the fibril axis, the elongation of the fibril axis run perpendicular to the stacking of the beta sheet. So the reason that we are interested in the membrane-associated fibro, well, one of the major reasons is that the generation of the A beta was naturally linked to the cellular membranes. As you know, the A beta was cleaved from the amyloid precursor protein, and two of the major secretases, beta and gamma secretases, especially the gamma secretase, is right in the middle of the membrane bilayer. So upon generation, the A beta was sitting in the membrane and then elute from the membrane and start to aggregate. And there are many evidence to show that the aggregation of the membrane causes disruption to different kinds of model membranes. And here shows the one of the previous results that when you add A beta to the synthetic lipid bilayer over time, cause the roughness of the membrane surface. And we also recently did some tests on the neuroblastoma cells. This is N2A cells from mouse. And you see that over time, you see the aggregation of the A beta on the plasma membrane surface. And the color here shows the co-localization of the A beta aggregation to the plasma membrane. And at very long time, you see the membrane contour become less clear and there are more aggregate forms. However, it is difficult and it's a really big challenging to clearly understand the molecular level mechanism of the membrane disruption caused by the A beta aggregation. The main reason is because there coexist of multiple possible pathways. The A beta could form different kinds of aggregates, and along each one of these pathways, there are different intermediates. So you can imagine that if you want to apply high resolution technique to this kind of system, they're going to be a mess. So over time, my group has put efforts on developing simple biophysical model systems. The aim is to develop model systems that leads to so-called predominant membrane disruption effects, which means within one model system, the A beta causes either membrane content leakage or lipid mixing or membrane fragmentation. And those different pathways has different end products. And the reason to do that is really to reduce the heterogeneity of the system and to make the high resolution study, make it capable to do the high resolution characterization. So today I'm going to focus mostly on the pathway that the A beta forms mature fibrils. Okay, so this model system involves the external addition of the A beta 40 peptides to preform the liposomes at relatively high initial A beta concentration. We try different concentrations range from 10 micromolar to 50 micromolar, and we see basically the very similar phenomena, the formation of the fibrils. And we found that the peptide to lipid ratio is a key parameter that changes the aggregation pathway. And this is one of the early studies to show that you start from intact liposomes, and over time, if you have different peptide to lipid ratio, at high ratio, like 1 to 30, in the end, you will see those fibrils. But if you reduce the peptide to lipid ratio all the way from 1 to 30 to 1 to 120, you don't see fibrils. Instead, you see those spherical oligomers. And the cyflavin T, which is the routine method to monitor the kinetics of the fiber formation, basically shows the same thing. At the high ratio, you see the buildup of the curve, but when you reduce the ratio, the fiber does not grow. And not only, so the peptide to lipid ratio not only affects the aggregation, it also influences the major membrane disruption effect. So when you reduce the ratio, you see the membrane content leakage gets suppressed. And at the same time, the lipid mixing get enhanced. So in either case, you only see those so-called local membrane disruption, either content leakage or the lipid mixing. You don't see major disruption to the contour of the lipid bilayer. So for the fiber formation, we're going to focus on high peptide to lipid ratio 1 to 30 from now on. we test different model membranes, and you can see here that the kinetic curves of the fiber formation actually varies a lot. So the membrane composition actually influence the so-called lag time and also the fiber growth rate. And for the structure study, we're using those biological membrane models. Those are isolated synaptic plus membrane extracted from grass bran tissue. The components are analyzed using the thin layer chromatography, and you can see it's fairly complicated. But when we check the high resolution structure of the fibers that's grown from different model membrane system, from the simple POPC bilayer to those RAS membrane, surprisingly, the MR structure feature, the cross peak patterns and the positions looks highly similar, which means that, and also the morphology that's shown on the TEM also looks similar. So that means there could be similar morphology and secondary structure for the A-beta 40 fibers grown from different model membranes. And not only the secondary structure, but also the tertiary structure, like those inter-residue interactions, also shows similar pattern for the fibers grown from different membrane. So we were wondering whether that's an indication that the membrane-associated fibers have some common structure feature that's actually different from the fibers that has been characterized a lot in aqueous buffer. So we picked this biological membrane as a model system, and we did high-resolution MR, try to generate a molecular level structure model for at least for the fiber core for this membrane-associated fibers. And here shows the negative stand TEM. One of the features that you will see is that those fibers you can easily form bundles, but there are also single filaments like that. And then we did the mass per length. It's a method to identify how many A-beta molecules are there within the cross-section of the fibril. And the result shows that the lowest number of the A-beta molecules is 2, but you don't actually see a lot of twofold mass per length, which will give you, if they're twofold, it will give you a mass per length around 18.4 kilodalton per nanometer. But the major peak really comes from the multiples of that, which is the indication that those fibers easily form bundles, as we show in the negatively stand TM. But for the unit, it's a twofold symmetry. Okay, then there's routine 2D spectroscopy. One of to identify the chemical shift, the backbone conformation generate from there. And one of the things that we saw is that at the C terminal, instead of those extended beta sheet that you really see for the A-beta 40 fiber, you see those kind of disrupted beta strand, like it's likely a short piece of beta strand. This is the inter-residue interaction that you, actually this is probably more clear. This is a summary of the inter-residue interactions that we found within the fibril. So there are side chain packing within one molecular, and there is side chain interactions with inter strand interactions here. Okay, and then another evidence is the detection of the interstrand distance comes from the PISART method. And so there are about 5 angstrom interstrand distance, which is an indication of the parallel in-register beta strand. And actually only short segments of the residues shows this close distance. The N and C terminals are pretty much far away from each other. And interestingly, the loop region is also kind of a little bit longer from what we usually expect for a parallel in-register beta strand. And this is different from the fibers that's formed in aqueous, which you always see like 5 angstrom interstrand proximity in the loop region, but you don't see that for those membrane-associated fibers. Okay, so overall, we put together a not very high resolution, it's kind of a medium resolution structure model for the core region of the membrane-associated fiber. This is from residue 16 to the residue 37, because the other residues, we don't see clear to the peaks are broadened out, means that they are probably disordered. But one of the features that in this fiber that's different from the essentially all the A-beta 40 fibers formed in aqueous is this kinked C-terminal beta strand region. And you can see the quaternary interface here. It's stabilized by the hydrophobic interaction formed by isoleucine 31 and valine 36, those side chain packing. But there's a kink around glycine 33. And we look at the literature and found that there's a previous published, this is not the structure model, but the backbone conformation of the A-beta 40 fiber grown from the PCPG bilayer is essentially show the same kind of feature, backbone feature. That this is a G33, the kind of forms a kink. And potentially there's side chain of the isoleucine 31 and valine 36 is pointing out, which means that it could hold a similar quaternary interface as we saw. However, there are differences too, like the side chain orientation of the isoleucine 32 and the leucine 34. They have alternate orientations. So it is possible that, because note that those fibers are grown from very different kinds of membrane models. So maybe this C-terminal kink is a common feature for the membrane-associated fibro, and it looks like from the side chain packing pattern that this could be a less tightly packed quaternary interface. So we were wondering whether this feature will somehow related to different, either physical chemical properties or the biological properties. So the next we check the stability of the fibro formed either in aqueous, the black one, or different model systems. And this is a guanidinium chloride dissociation of the fibros. And you can see that the membrane fibers are actually less stable to the denaturing. And noting that the guanidinium chloride is the water-soluble molecule, so it's likely that the water molecule is likely to more easily to get into those quaternary interface because of the packing here. And also we check the cellular toxicity using the N2A cells. This is only the N2A cells, and you can see that the black bar here is the aqueous fibro. Compared with the control that's only has the membrane. You see that the membrane fibers are generally more toxic compared with the fibros that's formed in aqueous. And we not only did those two, we did many different kinds of fibers grown from different membrane environments. In general, you see that the membrane-associated fibers are more toxic than the aqueous fibers. So that might also related to the dynamic of the ends, that those membrane-associated fibers are more easily to get those dynamic process of the elongation and dissociation process. Okay, so that's something that I want to show about the difference between the structure. But in addition to the final structure, we're also interested in intermediate states because we're looking at the membrane disruption in that process. And this is the plot of the lag phase. Remember, we did different kind of model membranes. It looks like in those simple lipid bilayers, the first 20 hours in our experiment, it's a lag phase. So in that period of time, it's kind of a nucleation process. The A-beta doesn't form fibrils. But when you test the membrane leaking, which is the major membrane disruption effect during this time, you see that the curve shows that the membrane leaking occurred like immediately. So within 20 hours, it's already in some conditions already get plateau. So the membrane disruption occurs way before the fibrillation of the fibrillation process of the A beta. So we go ahead and look at the time-dependent membrane association of the A beta, and we found there is within 20 hours, 50% of the A beta spawn to the membrane, and the population is growing over time. And these different colors indicate different membrane models. They basically show very similar pattern. Then if you check the CD on the membrane-associated A beta versus the A beta that's remain in solution, and what you see is that over time, and this is the initial state, that the membrane-associated A beta forms is kind of partially folded. It's not completely random coil. But when the time goes on, it becomes disassociated. And later on, it's assembled into a conformation that looks more like a beta strand. So there's a structure transition for the membrane-associated A beta within the lag phase. Now remember, this is still within the lag phase. There's no formation of the fibrils. But the peptide that's remaining solution, it's mostly random coil. There's no conformational change. So this makes us to think that in the presence of the membrane, really what happens is that the nucleation process is happening to the membrane-associated population of the A beta. There, they forms the certain kind of structure that's easy to elongate. Then they start to pick up the A beta peptide that's remaining in the solution and grow from the fibers start to grow from the membrane surface. And within the process, certain kind of structure change cause the local disruption of the membrane and the content leakage. So we are starting to use the DMP-based technique, and this is in collaboration with Professor Alexei Polipov, my postdoc colleague. He's currently a faculty at University of Nottingham at UK. We're starting to using the DMP-based technique to look at the intermediate states. We did some initial tests. The reason to do that was because two reasons. One, at intermediate states, only a small population of the A beta is bound to the membrane. So the peptide to lipid ratio will be even lower than the one that you actually add into the sample. And then because of the partial bond, the real membrane-associated A beta, the quantity of the labeled material, it's very low. So we test that using the DMP, we can get reasonable 2D MR with very, very low ratio, 1 to 150. I think we can get 1 to 200 peptide to lipid ratio with very small amount of labeled A beta material, like 0.2, 0.3 milligram. So it is optimistic to using this approach to look at the intermediate states. And we also get some very interesting results when we look at the time dependence effect of the membrane-associated A beta. So one of the things is that certain long-range interactions, like those side chain packing that you will see in the final structure, it already exists at very early stage. Initially when you add the A beta in there, that interaction already exists. Okay. I think I'm running out of time. I think it's 20 minutes or something. I just want to briefly show another pathway that we're actually looking at. This is the non-fiber pathway forming the oligomers, and this is some very recent results just briefly show that. So this is a different condition that when you go to very high A beta concentration, you actually don't see the fiber formation. You see those aggregates, and it's co-aggregates between the A beta, which is labeled in green, and the lipids, which is labeled in red in the confocal microscopy. And there are many studies show that those oligomers like to form membrane pore or ion channel. We test that, and what we saw is that the channel activity exists at the initial. However, when you start to incubate, the channel activity was less clear. Instead, you see like a overall disruption of the membrane for those large oligomers. So recently we developed a protocol try to extract those oligomers. Those oligomers actually stabilized by the lipids associated with it. We extract it using a protocol involves ultracentrifugation and SDS treatment. And in the end, we'll be able to collect the TEM imaging. This is the negative stand TEM imaging of those spherical oligomers. And remember, those oligomers have both A beta and the lipids in there. Okay. And it shows the interesting feature that when you dilute those oligomers, it forms individual spherical kind of morphology. But when you add them in really high concentration, like at the very beginning, it's a condensed gel. If you just do the condensed gel, all these things is kind of fused together to force a large aggregates, or even those linear-like but curvy kind of filament-like morphology. So we think that this is the future. This is the feature that highlights the presence of the lipids, because if you think about the lipid bilayer is like to fuse together. So when these oligomers have lipids around, it shows the feature of the lipid bilayer that like to fuse together at high concentration. And we did some initial AMR test on the molecular association between the lipids and the core of the A beta using the Riedel. And what we found is that there's a residue-specific interaction. So basically, all the residues that's located at the C terminal of the A beta and also the loop region, they're closer to the lipids. But the residues that's located that's closer to the N terminal is far away. And this is a very crude 1D spectrum. Shows that the C13 lines are reasonably narrow. So it is possible that in the future we can determine the lipid-associated oligomer structure using MR. I'm going to skip this. Okay, to summarize what we have, as I mentioned at the very beginning, that the membrane-associated A beta aggregation process, it's very complicated. And we were able to generate model systems that allow you to kind of separate those individual membrane disruption effect. And when we have an externally addition of the A beta to the preformed lipids at lower ratio, it forms the lipid mixing. This is something that I haven't talked about in this talk. And when at high peptide to lipid ratio, you form fibrils. And we see, early stage interresidue interactions. It induces rapid lipid membrane leakage. Eventually, it forms a structure feature that has the kinked C terminal, which could be a common feature for the membrane-associated fibrils. Now, when you have a high concentration of A beta pre-incorporated into the liposome, you see co-aggregates between the A beta core and the lipids that's surrounding it. So this is the target that we're going to using the high resolution MR to do some structural work in the future. So with that, I would like to thank the people that's involved in this work, mainly my graduate students, Xinhui Chen, and my postdoc, Lucas Cleese, and several undergraduates that make contribution to this work. And especially, I would like to thank my collaborator, Professor Polipov, who do the DMP work, and also the financial support from SUNY Research Foundation, NIH, and also the MRI program for the MR spectrometer. So with that, I would like to thank you all for your attention and be happy to take any questions.
Thank you, Wei, for the very interesting talk. So there are several questions in the Q&A. I'll start with Christoph Groha's question. Thank you for the interesting talk. Could you explain again how exactly you measure the interstrand distances? Maybe, I think that might be referring to the pi-thirds method, or?
Oh, so yeah, so interstrand distance is measured by pi-thirds.
Or
so this one. So it's a pi-search method that do the homonuclear recoupling. So you use the single labeled, like you put a single labeled residue along the one A beta strand, and when it forms fibro, all the labeling sites stacking together. So it really depends on the proximity between those different strands. If it's parallel, then it falls into like 4.7, 4.8 angstrom distance, and it gives the fast decay curve. And if it's longer away, like those over 7 angstrom or 8 angstrom, you don't see much signal decay. This is a I would say semi-quantitative way to assess the interstrand distances.
So Venus Sin Mithu has 3 questions, so I go one by one. Is it possible to know which species interacts with the membrane? Is it the monomers or oligomers?
Okay, so it really depends on different model systems that we're looking at. So let me see, for the external addition, we starting from monomeric peptides. We do pre-treatment for the A beta 40. And one of the reasons we use the A beta 40 is because it's easy to handle compared with A beta 42. So we ensure that it starts from monomer for those external experiments. For the pre-incorporation, I'm pretty sure it starts from large oligomers already, because the concentration is very high, and it's the way that we prepare the sample is that it forms lipid film. Right, it forms lipid film with high concentration of the A beta with the lipids. So as soon as you start the freeze-thaw process, I think the oligomers are already there.
The second question is, how do you make sure that fibrillation is membrane-driven and not happening in solution?
Okay, so that's related to what we have in the CD. So basically, during the lag phase, within the first 20 hours or 10 hours, the conformation of the A beta in solution remain random coil. So this is the clear evidence that you don't have nucleation process happen in solution. So basically, we think that this is the conformational transition happening on the surface of the membrane that drive the further aggregation.
Mm-hmm. Okay, that pretty much addresses his third question. So I'll go to Thomas Schmidt's question. And he asks, is it possible to have insights to the dimerization or trimerization or oligomerization process that occurs on a membrane once the monomer assumes some membrane-induced structure prior to fiber formation?
I think that's possible. So that's something that we're going to look at in the future using the DMP. That's not that's not something that we could do using routine MR, because the low abundance and because the even it forms dimer or trimer, I would think that it doesn't form a very well ordered structure like the fibrils. So we have to go to the DMP based high resolution technique to be able to answer that question. But that's that'll be my future goal.
So Carol Post has a question. The simultaneous mixing of protein with lipid is interesting. Do you think this is something that occurs biologically in cells?
Simultaneous means the pre-incorporation. Is that something? I think, well, assuming it's a pre-incorporation process, yes. So I think it's possible. So one of the reasons is because the local concentration of the A beta in membrane is not homogeneous. In some membrane region, the A beta concentration could be very high, especially I think there are previous characterization of the lipid rafts, which is one of the membrane region that could cause, aggregation, like local aggregation of the A beta. So yes, to answer the question, the short answer to this question is yes, some biological membrane location could have very high A beta concentration.
Okay. So Oleg Antukin has a question, does toxicity of membrane-assisted fibrils correlate with dissociation association equilibrium between fibrils and beta sheet rich oligomers?
I think that's a very reasonable hypothesis. We haven't tested that yet. Yeah, that's something that we're gonna test in the future, and this is related to one of my postdoc work. Like you have the difference between the twofold and threefold symmetry fibrils, where you can looking at the difference, the correlation between the end dynamics and the toxicity. Yeah, that's something we're gonna look at.
So Patrick Vanderwell has a question about terminal segment residues, which are missing from your ordered fibril structure, and he's wondering, are these residues flexible or disordered or membrane associated but not fully flexible? What's going on with these? Yeah.
We don't know yet. It doesn't show cross peaks in the solid state, so maybe we could do some kind of net-based technique or solution MR to look at this. Yeah, it's good to have a full structure model, but we don't have that information at this point.
So related to that, I sort of have a follow-up question. Do you think your dimeric species is actually intercalated between two lipid membranes? Because otherwise, how do you keep the symmetry of the N-terminal strand facing the lipids on one side and not the other? So any questions how this thing is sitting on the membrane?
So the question is about the membrane-associated fiber, how the quantum interface. Let me see. So one of the things I haven't shown here is that I think based on our understanding of the time-dependent formation of those inter-strand association, I think the C-terminal strand associate earlier than the N-terminal beta strand. So it looks like this is the region that's actually initially bind to the membrane. And it is possible that the reason that forms this conformation is because, it could be some kind of specific interaction to the membrane or the effect of membrane curvature that caused the backbone conformation looks like that. But how does quantum interface form? I think once it forms nucleus, then the fibers grow away from the membrane. Then because of the hydrophobic interactions, there's a preference that this interface would be associated with other A beta molecules that forms this kind of quantum interface. But that's a hypothesis.
I'll finish just one more question before we move on to the next speaker. So Ratiko has a comment, measurements of toxicity in cell culture generally show that soluble monomeric A beta has low toxicity when added to cell cultures. And your data show that membrane disruption occurs during the lag period before fibril appear. How do these observations relate to one another?
Can you repeat that? I'm sorry.
So in cell cultures, the measurement of toxicity will show that the monomeric A beta is less toxic when added to cell. And your data show that membrane disruption occurs during the lag period before the fibril occurs. So these two seem to be at odds, and he's asking how do these observations relate? Relate to one
I see. Oh, yeah. So the cell toxicity, well, first of all, the cell toxicity mechanism is not clearly understood. The membrane disruption could be one of them. I think the membrane disruption would be induced not by the monomer, but by certain kind of early stage small oligomers. So that could happen in the lag phase, but not the monomeric states. Is that answering the question?
Well, I think we can discuss after the second talk, an informal. Okay. All right.
Okay.
We'll move on to our second speaker.
Thank you.
So I think it's now my turn, right? It's my pleasure to introduce Rasmus Linser as the second speaker. Rasmus actually studied chemistry at the university in Göttingen and then decided to move to Berlin to do his PhD actually in my group. And this was in the time period between 2006 and 2010. And then afterwards, he moved on to become a research scientist in the Analytical Center in the University of Sydney in New South Wales in Australia, but then figured that this is maybe not sufficient, and he wanted to go back to research. And therefore, he picked up a postdoc position in the group of Gerhard Wagner at Harvard University. But this was also not interesting enough, because then he thought maybe he needs some changes, and then he decided to have a dual appointment, being a postdoc in Gerhard Wagner's group, but at the same time have a postdoc at the University of New South Wales in Australia, because this would increase the likelihood to be in Berlin, where his girlfriend was living at the time. So he had also a chance to visit her. In 2014, he returned back to Germany to become an independent research leader at the MPI in Göttingen, in Christian Griesinger's environment. In 2016, he moved to Munich, to the LMU. We had first professorship. And since 2018, now he's at the MPI in Dortmund. And today, Rasmus will talk about proton-detected solid-state NMR experiments and how he applies those to characterize the dynamics and the function of large enzyme complexes. So I'm very much looking forward for your talk. And yeah.
Thanks so much, Bernd. I have one little correction. It's actually the TU Dortmund that we're at.
What did I say?
MPI Dortmund. But we do have a lot of interactions with the MPI, and we're also part of the Max Planck research school. So we get talented students from that pool. So that's good. Yeah, let's just maybe start with the slides. Good. So as I said already, thanks to Bernd. This was very kind of you to introduce myself here and with these kind words. I'm also very grateful for the opportunity to present some of what we've been doing in the last years here. And I would have introduced where we are right now anyways also, which is in Dortmund. And that's something, that's a city actually in the very west of Germany that most people only know from its football team. But we did not move there because of the football team. Otherwise, we could have stayed in Munich. But instead for really a great research environment that we have there, including really great equipment, space, interaction partners, including the MPI people, for example. And so that's really a great opportunity, even though, of course, as you may have known or guessed from what Bernd has been saying, I mean, any move also has its pros and cons. But we're pretty happy there now. And I'm going to talk to you about NMR's big strength, or one of NMR's big strengths now, which is protein motion. And most of what we've been interested in fact, is microsecond to millisecond timescale motion. And that's something that has been a great topic, obviously, already in solution NMR. That's also, for example, what Dorothy Curran talked about pretty much last week. And we have, as ART has done really beautiful work on, for example, we have lots of techniques that correspond to relaxation dispersion methodology. And that's what these RD curves look like in solution, usually either CPMG or R1 row measurements. And in solid state NMR, they look slightly different. In fact, we do have the same regime. That's the Bloch-McConnell kind of relaxation dispersion, where we just basically have refocusing of the isotropic chemical shift differentially as a function of spin lock field strength in R1 row experiments. And we do have a second regime here, which is what we call the NERD, or the near rotary resonance relaxation dispersion. That's actually how Paul has been called, Paul Schander, whom we've had very many very constructive interactions with. Thanks, Paul. This work that we've been doing here together has actually been driven mostly by Petra Rovo, a postdoc in the group, very talented, and now a junior group leader in Munich, in fact. And this NERD regime is nothing else but basically the recoupling that we would usually have in a static powder. For example, if we have a magic angle spinning that means a certain rotation speed, a frequency, for example, in this case, in this simulation here, with 40 kilohertz, and then if we have a spin lock field strength of also 40 kilohertz, that means that we have exactly basically 280 degree pulses per rotor period, and that means we exactly recouple all anisotropic interactions. So the effect of the magic angle spinning, which usually averages these anisotropic interactions, is then vain, and we have a very fast decay of our, or effective decay of our coherences. So that's why we usually, for a static powder, we would have this kind of discrete jump here at the rotary resonance condition, but in the presence of internal motion, which can potentially happen on this order of on this time scale of the same time scale as the rotor period. That's usually on the order of 10 microseconds, for example, for 100 kilohertz spinning. Then we have an interference of these two motions, and then we can basically recouple already at an R1 rho of an spin-lock field strength that's not yet the actual recoupling condition. So we have a broadening of the condition, and that can be analytically calculated and traced back, or numerically simulated and obtained data from in very much detail. So that's what we would have from 15N nerd. And what we've been looking into in addition is proton nerd and protons, whereas 15Ns would basically recouple the CSA of the amide as well as the proton 15N dipolar interaction. The protons would also be able to recouple the proton-proton homonuclear dipolar interactions, and those would be recoupled here at the so-called horror condition. That's at half rotary resonance, for example, at 20 kilohertz when we spin at 40. And that has certain advantages and certain disadvantages. And one main feature here is what you see in this comparison. The 15N node really is an axis or a reporter on local dynamics. Whatever moves would give this kind of relaxation dispersion effect, and the rest wouldn't. And proton, since the homonuclear dipolar coupling network is perturbed or is recoupled, then we would also have reporters that are just close by the actual site of mobility, and they would also report on this. So that can be of advantage, but there are other drawbacks. And so that's basically just to introduce some methodology that we've been working on. And now I want to introduce a system to you, and we've been, or our major focus has been to just increase to make more associated and more methodology feasible to expand the scope of this technique and to make use of it and for more systems and more biological questions. And so all of this methods development part, usually we had been doing with the SH3 domain of chicken alpha spectrine. And at some point people were saying we were doing nothing else but SH3 research, which is obviously not true. But then we were looking for something just like SH3, just a little bit larger. And we found carbonic anhydrase, HCA2, as a good target that we focused on for quite some time in the following. And so this is actually when we were dealing with this kind of topic here, which is fast magic angle spinning. So for example, spinning faster than 100 kilohertz, and what benefits would this have in particular for dealing with fully protonated samples rather than deuterated and proton back exchange samples. We actually did this now, for example, regarding structure calculation or protonation states in side chains like in histidines, which you can read out from certain HN or HC spectra that you now have access to, as well as pKa values that you get access to from just J couplings that are specific to the various cases, as well as sidechain relaxation and dynamics. And we did all of this using the HCA2 new SH3 of ours, let's say. So when we did all of this, we noted that this protein was actually not rigid, opposed to what we were thinking it was. And so let's have a closer look. So this is the enzyme. It's basically a rigid core, or that's what people have been thinking, with a little funnel, an active site cavity, and in the middle there sits a zinc cation. And that is basically, or the enzyme in general is just catalyzing this interconversion between CO2 and bicarbonate. So that means that there is a hydroxide ion, anion, that sits here on the zinc and needs to be like correctly directed in such a way, or oriented in such a way that it can do a nucleophilic attack to the carbon dioxide and hydrate it. And then there must be a proton, which is basically then too much, needs to be channeled out of this active site. And yeah, this is basically this catalyst. It's an excessively studied enzyme. And there are still some open questions, however, which is, for example, what's the role of the conserved water network in here? Because when you look at different crystal structures, and there, as I said, this excessively studied, there are more than 700 crystal structures of this protein, you would see that the positions where the waters sit, they are very much conserved, which is due to the fact that there's a hydrophobic site here and a hydrophilic patch. And on the hydrophilic patch, there's lots of H1 donors and acceptors that would basically have a very specific ordering of these waters in the cavity. So that's been subject to a lot of research, and also something that we were interested in. And actually another aspect is that people have been thinking that H64, this is the proton shuttle that basically gets the proton out of this active site region into the bulk solvent, is the only moving part. And the rest of the enzyme was completely rigid. And this is, for example, represented here. So that's a textbook example of an enzyme with an extremely rigid pocket. This is just some quotes here. Carbonic anhydrase as an example for a rigid enzyme. That's a chapter in this book by Herbert Waldman. In fact, so that's a very well documented and often read book. This is another quote. The high rate of the enzyme is evidently due to the weak binding of the substrates, as well as the firm structure of its active site, the only moving part being histidine 64, and so on and so forth. There's much more. And this is all due to the fact that people have been looking at this only using X-ray and neutron diffraction. And in fact, the X-ray B factors are close to 0 all over the place here. And this is this active site loop here, which we will be looking at in more detail in the next slides. This is also very much close to 0. Then there is actually no difference whether or not there is an inhibitor here in the active site. This is again this active site loop. It's exactly the same conformation, and this is just the RMSD over the whole range of crystal structures, or in general diffraction structures that are out there. And in particular here, this active site loop, there's no deviation between these structures. So that's the picture of the X-ray. So that's actually 260 residues, or 29 kilodalton, I should say. That's why we chose this in the first place, to increase the size range or the molecular weight range of NMR, associated NMR at that time. And so when we were doing this water kind of research here, with most interest directed towards this water network, we actually found that looking in solution NMR, this active site loop was not rigid at all. So we have here R2s, and this is in fact this T199, which sits here at the very tip of the loop, as well as the T198, and the T198 is exchange broadened. So that's not been assigned previously, and we can show here by comparing solids and solution NMR that actually in the solid state we can nicely get this peak, and this is really unambiguously assignable. But in the solution case, it's absent, and so there must be some exchange going on. And we looked at this as we didn't see anything in solution. I mean, we didn't see that peak that was most interesting to us in solutions, so we thought let's just tackle it in solids in the first place. And what we did is we actually used these 3 kinds of methods that I introduced to you in the very beginning. This is the Bloch-McConnell relaxation dispersion, and actually what I should say here, in the very first regime here between 0 and 2 kilohertz, the solid state NMR Bloch-McConnell relaxation dispersion rates, they are not trustworthy because the spin lock field strength is just not sufficient to actually keep the coherent effects out of our measurements. So it's just not clean, let's say, dynamics data that we would see here. But everything after 2 kilohertz, that is where we see relaxation dispersion effects and trustworthy ones. And so this is actually the 15N BMRD. This is NERD, just in comparison to some prototypical standard, let's say, rigid residue. So you see that this is not, let's say, a baseline effect or maybe a global motion like the rocking motion that many has been an aspect of much research. So this is really only for these residues, and you see it, for example, here. And what's really interesting is this is representative for the active site pocket, this T198 now that we see in the solid state, and we also have rates, as you see in the next slides, also for the other active site residues here. The funny thing is that there are also dispersion effects on the other side of the pocket, even though there is no structural connection between these sites. And also the backbone amides do not show any dispersion, but it's only these amides here that are actually in close conjunction to this conserved water network. And an additional effect is that actually this dispersion can be quenched by putting in an active site inhibitor. And as I said, this is these coherent contributions, so that's something that you can neglect, or even if you fit it, this would be a totally different regime. Usually this would be all fit to the 100 microsecond timescale regime. So, and this again the same for this active site loop, as well as these sidechain amides here. And so the question was, how could these be coupled to this movement? If something is moving here, is this maybe the water? Is there some effect of the water, in fact? And we looked more in detail into this water topic. And maybe some other information that you need to know to understand what we've been concluding from this is that there are actually not many very drastic perturbations if you compare crystal and solution NMR on this target. I'd say there are some that are very, very strong, but they can be traced back to crystal-crystal contacts, and the others are just on the order of, let's say, 0.1 ppm or so, as expected. But there's one thing that's really different, which is actually the N67, this aspergin that we just looked at. If you compare the free form with the in solution with the free form in solids, then it's this H delta 22 that moves by almost roughly 2 ppm. So that's a pretty large chemical shift perturbation, and the same actually happens when you look at the free form compared with the inhibited form, even though this inhibitor is not in contact with these residues directly. So this cannot be a ring current or something, or some other direct perturbation. So that's really something that must be due to this environment here, which somehow has to do with the water. And so actually, if you put the dozolamide or acetazolamide, other inhibitors into the solid sample, then there's no further shift. So there must be some kind of two different situations here on average for the environment of these sidechain amides. And actually we looked into what we would expect from just water or differential H bonding to water. And we did some calculations on the basis of DFT. And this is basically just qualitative. So whenever you have a water that's close to some H bond amide aspergin sidechain, for example, then and if you move it closer or further away, so dependent on the angle and also in comparison to implicit water surroundings, you would have effects on the order of 1 to 4 ppm or 0 to 4 ppm of perturbation, in fact. And this is just showing here an example where we take the crystal structure and let it relax into a different position. So that's what it would spontaneously go to. And this different position here already makes it 2 ppm, depending on how you calculate it in detail, around 2 ppm perturbation. So is there something that has to do with the water environment? And in order to really clarify what was happening here, we turned to MD simulations, and the MD simulations showed us what actually is important to notice, that the water itself has a lifetime on the order of only a nanosecond or a few nanoseconds, but it hops always back into the same conserved positions. And that's very much in conjunction with what we know from the crystal structure positions already, even though there's a slight difference because after the MD simulation starts, it would basically relax a little bit into a slightly different conformation, but then it would be very well defined spots for the water. So that's the blue cages here where the water sits with, I don't know, 95% or so probability. Good, so we have very fast water, but it jumps back into the same places. So what we think is actually happening here is that we have some kind of motion at the very active side, and then we have a water which is in a certain kind of geometry, in a certain kind of architecture, which gives a certain kind of situation here for these residues. And when this moves into its excited state, for example, which is this 100 microsecond timescale motion, then we would have a differential situation of the waters. They would be somehow different. We don't know what exactly, but somehow different. And this would then mean a slightly different average surrounding chemical environment here for those, meaning a different chemical shift. And this would then give rise to a concerted, a collective effect when we have a chemical shift, a relaxation dispersion effect here. We would see that also here through the correlating effect of the water, which is really just a very indirect way of transferring the relaxation dispersion. But we still don't know, or at this point, what is that 100 microsecond timescale motion? And we looked a little bit deeper, and we turned back to solution NMR, and we get, if we just heat it up to a little bit more temperature, for example, this is 45 and 37 degrees Celsius, then we can also see, for example, this T198, it comes back, but with a very low signal intensity, but it's enough to actually get relaxation dispersion rates. And what we see is really the loop has relaxation dispersion. This is actually increasing, the phi x is increasing here to the very tip of the loop and then goes back down. So that's moving. And then we also have this same relaxation dispersion at the N terminus, as well as the this proton shuttle. So actually the H64 is overlapped, but the G63 also shows this relaxation dispersion. And this can be all globally fitted. And the same is actually true if we do this for solid state NMR. So we get basically the same, qualitatively the same, as just that we would globally fit this to 204 microseconds. Now this is different data. Whereas we are only at 30 degrees Celsius here and a 1.3 millimeter rotor, 55 kilohertz spinning. So this is, let's say, a little bit faster, but qualitatively it's the same. So what's also important to note is, again, when we have this inhibitor in here, our relaxation dispersion ceases, and this is just a representative. Here, that's the same for the rest of this loop, but it does not cease. So from here to there, this is with and without inhibitor, and this is with inhibitor, but it does not cease when we compare this G63, which also in the inhibited form has relaxation dispersion, however, with a slightly different time scale. So this is the overall plot of a sequence. So this active site loop is quenched, and then this proton shuttle is actually not quenched. And the N terminus, unfortunately, we don't know exactly. We think that there's still some dynamics going on. These peaks are still very weak, but we cannot really get conclusive data out of this. Good. In order to really approach that a little bit more mechanistically, we thought, okay, let's try solution NMR structure calculation. And we calculated a structure here. And unfortunately, right in this active site region, the RMSD, the convergence is not really high. It's like 1 angstrom RMSD. So it's not, let's say, totally bulletproof, but we see in the average structure, there is a difference to the crystal structure, in particular this loop. So this is the RMSD over sequence with respect to the crystal structure, and this is this active site loop. Maybe a little bit more informative is also the RDCs when we measure those and compare them with the theoretical ones in Marco's program. Then we actually see a lot of these residues in the active site that are not what they would be expected to be. But again, this is maybe not yet totally waterproof, and we actually turned again to MD simulation with our collaboration partner Lars Schäfer. And here in this case, unfortunately, in order to get this microsecond, this is a 100 microsecond timescale motion, in order to get this into an MD simulation, which usually is only a nanosecond, this is, or a few nanoseconds, this is something that we had to do. We had to actually truncate this protein in order to make space for things to happen here. And in fact, and just basically in order to see the mechanics behind this, because the experimental proof that something is happening we get from the NMR. And in fact, this enzyme is still, even with the truncated N-terminus, is still active. It just has, I don't remember the exact number, but it's like a little bit less active. I don't know, 10% less active or something, but it's still pretty much physiological. And in this case, we see that, in fact, the ligand bound form has a very defined position, whereas the free form has a pretty much lesser defined position. And what happens is actually that this hydrogen bonding situation here to the zinc can be detached, and there is kind of an active side open-close motion. That's possible, at least mechanically. And so then the N-terminus somehow plays a role, but as we said, or as I said, this cannot be, there's an end to the detail that we get into this. Good. So as the last topic, I'm running out of time here already, is that actually I said we wanted to actually, our major aim is to actually push the boundaries for solid-state NMR and maybe NMR in general. And so 40 kilodalton or 30 to 40 kilodalton should not be the limit. And we've been looking very much with much effort into a protein that we've been tackling together with Len Mueller from UC Riverside. And it's tryptophan synthase, and this is a 2 times 72 kilodalton or 2 times 650 residues protein. So that's only the asymmetric unit, basically. And this is a pretty big thing to look at, and we have been able, with really lots of efforts, to get on the order of 60% of really completely backbone and C beta assigned residues, and in addition we get some that are out of this selection that are partially assigned. So we have quite a substantial pool of residues that we can now deal with in order to apply different kinds of techniques to this. And in order to get there, in fact, we actually had to go to higher dimensionality, because our 3D methodology and even this year would not have really worked. Or let's say in general, the trend is, if we have higher dimensionality, of course, we get better dispersion in the peaks. And this is 2 new sequences that we came up with. Most importantly, a 5D HNC ACONH, and the sidechain to backbone experiment that correlates in a 4D fashion all the sidechain carbon shifts with the amide HNCA. Good. So with this in hand, with, let's say, the magic hands of Len Mueller's group, they could actually turn this into a nice NMR crystallography study. And for example, here, this is this sidechain to backbone experiment strip for one of the most important residues in this protein, the beta lysine 87. And actually, it's been of much interest because it carries this cofactor, the pyridoxal phosphate. And there's been lots of debate about whether this is a protonated shift base or a deprotonated one. And in fact, now with the chemical shift values that we have for this, Len could actually set up different models and calculate the expected chemical shifts, and from this really calculate what is the, let's say, the thermodynamic situation here of this shift base. And another thing, which obviously also fits to the remainder of the talk, is that now we have an access to relaxation rates. In this case, this is R1 row relaxation rates, because really relaxation dispersion would have been too much in terms of measurement time for this beast. But even the R1 rows that we can qualitatively interpret as the presence of millisecond to microsecond timescale motion, this is really interesting data already. And as we see here, this is, for example, the rates in the beta subunit active site, and there's lots of stuff going on. For example, here at this site, we have rates up to or around 9 per second, and here also there's it's much larger than, let's say, the baseline that we would expect for rigid residues, which you can see here. It's like 1.5, 1.8, something around this, 1.2. And it's really those residues that are expected to be perturbed whenever there's a change from the closed to the open state of this entry portal for the serine into the subunit. And in crystal structures, this has been expected to be just very well defined states, dependent on what kind of substrate you put in or how you stabilize a certain state of this catalytic cycle. But in this case, we can really see that there is spontaneous motion, spontaneous fluctuations going on at room temperature on that time scale. And so that might well be linked with a global conformational exchange or global processes that have been of much interest in tryptophan synthase as the allosteric communication between the alpha and the beta subunit. So this is really very interesting research in general, and it's really important to know that for these kinds of proteins of this size, people would not really have thought that such NMR details would be accessible. With this, I'm actually done with the talk, and I would very much like to thank the people that actually did the work. And I have mentioned, or at least shown, pictures of the people that have been, of whose data those were that I showed, and this people that are here in red. But of course, I'm very grateful for all the members of the group, of the current group, as well as the past members in the group over the years, as well as the collaboration partners that I mentioned, or actually Peter Günther for. So all of this assignment was done after initial trials in manual assignment, but with 650 residues, we thought, okay, it's more intelligent to actually do this with more intelligent algorithms, and Peter Günther actually turned this into something more useful. Of course, funding and past interaction partners in these various institutions that I'm very grateful for. And not the least, of course, I'm grateful for your attention and interest in this talk, and I'm happy to take questions.
So thanks, Rasmus, for this excellent talk. There are a number of questions in the chat, and the question and answers. So I will ask also the questions, right? Okay, so there's an anonymous participant who asks, why is the initial regime in the dispersion data that is not trustworthy not considered in the numerical simulations? What is missing in the theory? Maybe Rasmus, you can.
Well, I mean, this is a little bit difficult. I mean, that's maybe also not my main expertise, I would say. So if you do these, I mean, the calculations as you would fit them, this is really based on the Bloch-McConnell equations, and that's basically not having any coherent interactions that can be treated in this algorithm. That's something that's really typical or important and unique for solid-state NMR, and it's really something that people don't understand in all detail. I would say what's these contributions to the apparent rate. You basically just try to spin those contributions out by as much as high as possible magic angle spinning rates or deuteration, or in order to get rid of these direct interactions. But in the sense of not having enough 15N spin log field strength, basically what this goes into is the T2 instead of an R1 row, and this is very much more perturbed by these coherent interactions than the R1 row decays.
Okay, Patrick van der Wel asks, you seem to say that the X-ray B factors gave no indication of active site motion. Do you now think this is because the X-ray was done at low temperature, or is it a crystallization artifact?
Well, that's a difficult question. I guess that usually the X-ray B factors, they get higher for higher temperature structures. But I guess for this, no one has, I'm not actually sure if there are room temperature structures also. But the thing is that the B factor itself is just not a very good measure for this kind of motion, because it would always reflect, I mean, the populations as well, and not very sensitive to, let's say, low populations of excited states.
Well, in the same sense, in the same sense, you saw differences between the solution state NMR data and solid state NMR data. Like when I think about this one asparagine, which was only visible in the solid state. So there seem to be differences in the dynamics of the crystalline state and the solution state, right? And this might be due to temperature, but it could also be due to crystal packing effects that affect local dynamics.
Yeah, that's true. However, we really think that the dynamics in the solid state NMR that we see from solid state NMR are very much the same as in solution NMR. I think there's a shift in time scales, and obviously very large rearrangements cannot happen in a crystal. But whenever something is not, let's say, completely forbidden due to crystal packing, then you still have the same tendencies also to happen within the crystal. But the B factor is just not a very good measure, and people were maybe really also looking only at cryogenic structures. And so this really just emerged as a very global misconception of the nature of the human carbonic anhydrase as an enzyme.
So also Elias Akuri has a question to this direction. Is the change in the chemical shifts of asparagine 67 pH dependent and influenced by the dynamics of the neighboring histidines?
Yeah, I mean, we did not obviously try any pH titrations, but I don't think there would be much of a shift, or if there would be a shift, then it would be a rather not a two-state kind of behavior, which we've been observing. So we've really observed this one and the other chemical shift. And I mean, differences in chemical shift just due to the, let's say, differential pH, for example, solids versus solution, that can be ruled out. It's been very clear that this is, or very, I mean, the postdoc Himanshu has been doing this with exactly the same pH in both aggregation states. And obviously, when a ligand comes in, then this is usually also not. I mean, we have all these buffers pH calibrated, and this only has some DMSO. It should not have such a large effect.
Emmanuel Verutis is asking, how can you know that the fitting function is appropriate when the error bars are so high? That is in page 15 for some of the dispersion curves.
Yeah, I guess, yeah, we're basically limited by the data, and I guess we can just do as well as we can. But I think what really proves all of this to be true is that we have these effects in all of these dispersion curves for different kinds of experiments, as well as in these also in the solution NMR experiments. So as I said, that's very much the same. And even if the error is so high, we would still basically, on the qualitative hand, we would be very sure that this is in fact happening.
So Marcus Cardoso asks, it seems that the nanosecond timescale movements of water molecules are important. Would it be possible to study larger proteins using this approach?
I mean, I mean, relaxation dispersion experiments can obviously always been done as long as you can assign your protein and have enough signal to noise. MD simulations, I would say we haven't really reached the limit for molecular size at all. So there are much larger proteins being studied by MD, and basically the data that I showed about the hopping characteristics of the water, that was mainly an MD kind of result. So I wouldn't see why one wouldn't be able to do this in larger proteins.
There's another question concerning the relaxation dispersion curves, and you mentioned that the first 2 kilohertz of the curve is not trustworthy. Would additional fast small amplitude motion extend this 2 kilohertz to higher fields? And how would you decide when to start trusting the data?
I mean, I would not expect this to be increased by higher amplitude or faster motions, because this is really a contribution from static direct interactions. It would scale with the density of protons, for example, or the size of the dipole. But if you have faster motions, they would rather average these out. So that's then a little, I mean, it would actually get better if you had faster motions in addition, I would think.
David Coburn says, great talk illustrating how well cavity fluctuations in enzymes can be defined. what are the prospects of going to much faster processes of active sites, resulting in reduction of activation energies and closing in on transition state structures?
Ooh, question. I have to see it again. And this is jumping all the time. So I guess if we have faster processes, then at some point relaxation dispersion would not really work anymore, unfortunately, even though with the NERD approach, we can go to much faster timescales than with the Bloch-McConnell relaxation dispersion. Just due to the fact that we have just way higher power, but if we really exceed this regime and can only get, let's say, fast timescale picosecond to nanosecond motion, I mean, then probably the excited state and activity how this influences the activity is not really described by conformational exchange processes anymore. And then yeah, I guess in order to really get structural or mechanistic data out, it also gets MD back into a better position, but I guess really faster motion, that's then a different topic. Yeah, and there's always this limitation of really translating, let's say, the order parameters into some kind of structural picture that one gets, and in our hands, MD has been very successful of doing this.
Okay. Do you see NOEs to water for active site residues? What restraints were used for the solution state structure, or for the solution structure?
Yeah, NOEs, I don't remember exactly anymore. There are NOEs to water, yes. Yes, but we could still not really rule out. That's the thing. I mean, whenever you see water NOEs, then this could also always be due to hydroxyls that are in some surface residues, which would just have a much longer lifetime, and as such, basically take the same role as a water that has a long enough lifetime to give rise to an NOE. Constraints of the solution NMR structure, I don't really remember, but it's definitely RDCs, lots of NOEs, Taylor's N, backbone dihedral angles, some H-bonds, sorry, H-bonds that we determined. Yeah, that's it. So it was pretty well refined, but unfortunately for certain regions, then the number of restraints were lower than, let's say, for the very well defined backbone regions here, or beta sheet regions.
There's one more anonymous question. At some point, you mentioned that for synthase, for tryptophan synthase, you had to measure 100 times more than for SH3. Could you elaborate on some of the experimental measurement time?
Sure. So for tryptophan synthase, yes. So that's maybe a general thing to mention. So whenever you have a protein that's 10 times larger, then I'm not actually sure if I said it, but you had to it's here, yeah. So it's 100 times more. It's a factor of the factor of the molecular weight squared. So this is really a lot of measurement time that we spent on this over 2 years, in fact, and we have all kinds of data, lots of very different experiments. And particularly these very insensitive experiments we recorded for multiple weeks.
So I think we come to the last question, at least in the chat, from Ryotaro Okabe. Thank you for a great talk. Can the spinning of the rotor affect the dynamics of the water in the sample robustly?
No, I mean, these are dynamics on very different time scales, I would say, and then the only effect that the rotor, the magic angle spinning, would have is a different different centrifugational situation, but this doesn't or higher pressure, but this is not really what would affect the water behavior significantly. Or I could not imagine why it would. I don't know if that answers the question. I don't know.
Yes, so I think we are through with the questions from the chat. So I would like to thank you, Rasmus, for talking, and maybe hand over to May again for final last words.
I think our formal session is over. Thank you for staying, and we'll see you next Thursday.